This video is about cell structure and functions of organelles, and we are going to learn 5 different types of organelle, which is mitochondria, lysosome, ribosome, chloroplast, and centriole. So from this diagram, mitochondria is the orange organelle. Second, lysosome is this round spherical organelle. Ribosome, which can be found on endoplasmic reticulum. Centriole, which is the zone which we can find centriole on it. And chloroplast, which only can be found in plant cell. So for the first organelle, mitochondria or mitochondrion, it is a rod-shaped structure that vary in size and the shape. So the size is about 2 to 8 micrometer in length. So mitochondria usually have their own DNA. So this DNA, they contain several genes that produce some protein for the role for the mitochondria to undergo cellular respiration. So mitochondria usually can be found in the cell which is metabolically active. For example, skeletal muscle cell, sperms, liver, or pancreas. Mitochondria has 2 layers of membranes, which is a smooth outer membrane and highly folded inner membrane called cristae. So the diagram of mitochondria, the outer membrane, which is smooth layer, inner membrane, which is highly folded. So if we look from the 2D structure, the highly folded inner membrane, you call it as cristae, while outer membrane is smooth. So the space in between the outer membrane and inner membrane, you call it intermembrane space. And the center of this mitochondria, you call it as a matrix. So this matrix is something like the cytoplasm of the cells, where you can find ribosome and DNA inside the matrix. Okay, so functions of the mitochondria is the site of cellular respiration. So cellular respiration mainly undergo inside the mitochondria to produce ATP energy. Okay, second organelle, lysosome, is a small vesicle formed when a small piece of Golgi body pinch off at the end. In other words to say, lysosome is formed from Golgi body. So inside the lysosome, it contains hydrolytic enzyme which used to digest the material within the cell. So lysosome is only single membrane bounded, but the hydrolytic enzyme inside the lysosome will not digest the lysosome itself. So lysosome involved in intracellular digestion. Intracellular means inside the cell. So inside the cell, digest the food material, it depends on the lysosome. It involved in the phagocytosis, cellular process of digestion. So from the rough ER, the ribosome produces the protein. Ribosome produces the protein and then it will be sent to the rough ER. Then the protein will be packaged inside the transport vesicle and it will be sent to the Golgi body. So Golgi body will help to repackage to become the hydrolytic enzyme, and then it buds off and then become the lysosome. So lysosome will be involved in the process of autophagy or intercellular digestion. So lysosome will fuse with the food vacuoles. Food vacuoles is formed when the process of phagocytosis occurs. So phagocytosis will take in the food granules and then it will be packaged into a food vacuole. So now the lysosome will fuse with the food vacuole and release the hydrolytic enzyme into the food vacuole to digest the food. So functions of lysosome are autophagy. Autophagy means lysosome release enzyme to destroy or engulf the worn out organelles in the cell. For example, mitochondria. So when mitochondria is all worn out, then the lysosome will fuse with this mitochondria to destroy the old mitochondria. Okay, so this means autophagy. Second function is to digest the material which has been taken into the cell by phagocytosis. So this is intracellular digestion. The third function is to release enzyme to outside of the cell to digest other cell in a process known as exocytosis. And the fourth function is autolysis. Autolysis means it breaks down the cell itself to digest the entire cell. Self-destruct. For example, the tadpole tails. So when the tadpole grow into the adult frog, the tail disappears. So the tail disappears because of the lysosome digest the tail. Okay. So autophagy means break down the worn out organelle. Autolysis means it breaks down the cell itself. And also this is the intercellular digestion. Okay, ribosome. Ribosome is a small or dense particle found in huge number in all cells. It is made up of rRNA and also protein. So ribosome consists of two subunits, which is large and small subunits. We have two types of ribosome, which is free ribosome and bound ribosome. So bound ribosome can be found on the surface of the endoplasmic reticulum, especially rough endoplasmic reticulum, RER. While free ribosome, it is just suspended in the cytoplasm itself. So the size of the ribosome, it is about 20 nanometer in diameter in eukaryotic cell, but slightly smaller in prokaryotic cell. So most of the protein made by free ribosome function within the cytoplasm, while bound ribosome generally make the protein that are either for insertion into the membrane or for export from the cell. So the two subunits of the ribosome, large subunit and also small subunit. So just now we say that eukaryotic cell has slightly larger ribosome size than the prokaryotic cell. So the size of the ribosome is 80S. S stands for Svedberg. Svedberg is the unit where we measure by the sedimentation coefficients. Prokaryotic cell, the size is 70S. So both the prokaryotic cell and eukaryotic cell, they have large and small subunit of ribosome. So for the prokaryotic cell, it is made up of 50S plus 30S, while eukaryotic cell is 60S plus 40S. So the main function of the ribosome is a site of protein synthesis. Chloroplast. Chloroplast can be found in plant cells only, not in animal cell. So chloroplast is a green color plastic contain the green pigments. Okay, so chloroplast contain the chlorophyll, which is essential for the process of photosynthesis to occur. Chlorophyll can also be found in eukaryotic algae, leaf, and other green plant organ. So the structure of the chloroplast, it is about 3 to 10 micrometer in diameter and 2 to 3 micrometer in thickness. The shape of chloroplast is biconvex, which is lens shape. So similar to mitochondria, chloroplast also enclosed by double membrane, which is separated by a narrow intermembrane space. So inside the membrane is a fluid-filled space which we call stroma. So inside the stroma, we have interconnecting flattened membrane-bound sac called thylakoid. Okay, so the double membrane of the chloroplast is smooth, not like mitochondria just now. Mitochondria has outer membrane smooth, inner membrane highly folded, But for chloroplast, both of the membranes also smooth. So inside the chloroplast, you have something like the cytoplasm, you call it as stroma here. Inside the stroma, you have the membrane sac, which is thylakoid. So thylakoid will be stacked up to form grana. Okay, grana. Grana is plural. Singular, you call it as a granum. So in between the double layers of membrane, we have intermembrane space. Okay, so chlorophyll pigment can be found on the thylakoid membranes. Main function of chloroplast, site of photosynthesis. Centriole. Centrioles are found in animal cells. It is about 150 nanometer in diameter and arranged at angles to each other. So centrioles are two short bundles of microtubule. They are at position at right angles to each other. They are just located outside the nucleus in a clear cytoplasm called centrosome. So centrosome matrix is a region where you can find centriole in the center. For animal cell, you have centriole. But for plant cell, they only have centrosome without the centriole. So the wall of each centriole is made up of 9 triplets of microtubule arranged at an angle. The wall of each centriole is made up of 9 triplets of microtubules arranged at angle. So when the time of cell division occurs, example mitosis, centriole will migrate to opposite poles of the cell where they produce the spindle fiber. So main function of the centriole is organizer of spindle fiber and involved in the separation of the chromosome during the cell division. In some of the cells, centrioles divide to produce basal body from which flagella and cilia develop. So this is the basal body, where below it we have the centriole. So for the structure of the centriole, it is made up of 9 triplets of microtubule. So each of the microtubule arranged as triplet, 1, 2, 3, 1, 2, 3, and then they arrange in 9.